Well, thank you very much for your invitations. Sorry for the invasion between the two presentations. Thank you, Lucille, for that. I need to go back to Paris this afternoon for the ceremony of retirement of my boss, but I will come here tonight. Sorry about this. So I will show you on the first slide on the heterogeneity of the arterial system and the pulsatility characteristic of the circulations. From the central artery to more distal artery, there is progressive reductions of extracellular matrix and the number of musculoelastic properties, together with an increase in the density of vascular smooth muscle cell at the periphery. And large arteries are two main functions. The first one is to deliver blood flow from the heart to peripheral tissues, and the second one, which is the cushioning function, is to transform the pulsatile flow into a steady flow at the capillary level. And large elastic potential have a large elastic potential, whereas muscular arteries at the periphery play an important role in vasomotor tone. And in fact, there is something very important, is that vascular smooth muscle cell from a specific site may influence the cushioning function, the function of arterial compliance. One vascular smooth muscle cell at a specific site may influence arterial stiffness, and another site due to the coupling between macro and micro circulations. It is relatively easy to measure arterial stiffness in large arteries. It is a little bit more difficult in muscular arteries. We need arterial sample in human, for example, or specific ultrasound parameters to be enough resolution to be able to measure simultaneously variation in arterial diameter and variation in blood pressure in the same time. So an important thing also is the existence of a pressure gradient between the blood and between the adventitia in the outer of the vascular wall, and there are 120 millimeter of mercury in the blood and 12 millimeter of mercury in the interstitial compartment, and these create an outward convection of many molecules. For example, prothrombin, for example, for the formation of thrombosis within the vascular wall, and these represent one of the main mechanisms of common vascular diseases. Arterial compliance can be defined with this slide. During systole, the arterial wall serves as an elastic reservoir of blood, and during diastole, arterial wall is able to rescue the blood flow during the diastolic part of the cardiac cycle. And these properties of the vascular elasticity define the level of arterial compliance. Arterial stiffness, which is a mark, the main characteristic of vascular aging. There is no vascular aging without arterial stiffness. So arterial stiffness is the proxy of vascular aging, coming from well-identified cardiovascular risk factors, gender, blood pressure, lipid, smoking, diabetes, but also and with less well-identified cardiovascular risk factors, from oxidative stress, for example, to genetic, epigenetic, and fetal reprogramming. And arterial stiffness, which is defined as the measure of arterial elasticity, is an integrator of all damage done to the arterial wall. And one of the easiest measurements of arterial stiffness is pulse wave velocity, which is the propagation time of the pressure wave along the arterial tree from the common carotid artery to the femoral artery, for example. And its definitions and its measurement require simultaneous measurement of pressure at two different sides and the time delay between these two measurements and the distance and the length between them. There are other arterial stiffness parameters. Pulse wave velocity is a general parameters of arterial stiffness, whereas we can also have local parameters of arterial stiffness, such as distensibility, for example, which were present at the level of the carotid artery, the relative distension of the diameter on variation of arterial pressure during the cardiac cycle. Another parameters is the elastic modulus, incremental elastic modulus, which is in fact is the slope of the stress-strain relationship, and arterial stiffness as related to incremental elastic modulus increase with the level of wall stress, and the real wall stress transmit to the arterial wall is not blood pressure. It is a combination of blood pressure, arterial thickness, and geometry of the arterial wall. There is a recent concept which emerges in arterial stiffness, is the concept of cellular stiffness of vascular smooth muscle cell. And with the force atomic force microscopy, it is possible in living cell to measure cell topography within the membrane of the cell, adhesion forces between cell and extracellular matrix with the environment of the membrane of the cell, and Young elastic modulus, which is an index of arterial stiffness. And we can see here in Young and old monkey the acto-myosin proteins, and you can see that the acto-myosin protein organization of cytoskeletal protein is much higher, the staining is much greater in old monkey compared to a young monkey. So there is a complete reorganization of cytoskeletal within the cell. But at this time, we don't know exactly the contributions of cellular stiffness to arterial stiffness at the level of the whole arterial tree. And we can see here the distribution of force, which is much higher in the old monkey compared to young monkey. Arterial stiffness has demonstrated an independent predictive value for outcomes in various populations, in cardiovascular and general events. For example, total mortality, cardiovascular mortality, hypertension, stroke, coronary events, but also in general populations, elderly, hypertensive, diabetics, and chronic kidney disease. One of the most important disease in which arterial stiffness is the highest is chronic kidney disease. 3 slides to show very rapidly that there are many guidelines, recommendations coming from European Society of Hypertension, of European Society of Cardiology for the management of arterial hypertension and the management of arterial stiffness. And for example, from 2013 to 2016 and now 2018, there is modification in the recommendations of arterial stiffness useful to determine the cardiovascular risk. And arterial stiffness is necessary to address the cardiovascular risk. And in the last guidelines, measurement of pulse wave velocity may be considered for measuring arterial stiffness with level 2B and class B. But it is not very important to see at this guideline, sorry for the expert of these consensus, because the most important things, we should not to look only at these recommendations. Between the questions is to find the best marker, the best parameter able to measure the cardiovascular risk in the overall population and specific populations. We know now that the normal blood pressure decreased more and more. Before it was 140 millimeter of mercury, now it is 120 millimeter of mercury, and probably probably very rapidly all of us will be treated for hypertension and all cardiovascular risk factors. So this is a very real problem, and we don't know at this time which is the best parameter between blood pressure and pulse rate velocity, for example, arterial stiffness, which is the best to understand cardiovascular risk factors. We know, for example, in the very old aging, that it is almost impossible to measure correctly blood pressure. So now we know that the most important cell population for arterial stiffness is represented by vascular smooth muscle stiffness, by vascular smooth muscle cell. And smooth muscle cell plasticity is explained by different functions. They are represented here. And it is important to note, for example, that common characteristics are very similar in different vascular diseases. For example, pro-inflammatory phenotype, extracellular matrix synthesis degradations is common to different vascular disease, for example, aging or atherosclerosis associated with firm cell. We know that calcification and firm cell are common to atherosclerosis. We know that contraction, relaxation changes, some degree of dedifferentiation of vascular smooth muscle cell and senescence are common to hypertension, and also that proliferation and migrations are the main events producing antimal thickening, which is one of the earliest common feature of common vascular disease. So I would like to recommend this recent spotlight in cardiovascular research, and they try, the authors in the series of papers, they try to make a new recommendations, a new classification of vascular smooth muscle cell properties. And the topic discussed in this spotlight issue could lay the groundwork for smooth muscle classification based on the diverse origins, that's important, and fate of vascular smooth muscle cell population coming from various transcription, epigenetic regulators, and the typical smooth muscle markers. And the traditional classification is based on function in red and phenotype in blue. For example, contraction relaxations define the phenotype of contractility, whereas the function of repair, migrations, proliferation, extracellular matrix synthesis define the synthetic myofibroblast-like and mesenchymal stem cell-like phenotype. In addition, smooth muscle cell functional diversity includes also weakening, which is defined by extracellular matrix degradation, more particularly elastin network, stiffening of the arterial wall, hypertrophy, and cell death. And to go further, in the role of vascular smooth muscle cell in vascular aging, we need to make an integrations visualization of all the vascular properties of vascular smooth muscle cell. And they are classified into different functional families: matrix, hemodynamics, inflammations, contraction, mechanosensing, transcriptions, genetic and epigenetics. And all of these functional properties define the vascular smooth muscle plasticity involved in vascular disease and arterial stiffness. And I will focus the second part of my presentation of mechanosensing in relation with hemodynamics and genetic and epigenetics. The paradigm of age-related arterial stiffness has recently shifted from elastin collagen content, increase in collagen, fragmentation of elastin network, to cell extracellular matrix interactions and vascular smooth muscle tone as key molecular cellular determinant of arterial stiffness. And we can first look at the integrin, which represent major receptors involved in mechanotransductions in vascular smooth muscle cell. And if we look, for example, at alpha-1, beta-1 integrin receptors, which binds collagen, we can see in invalidated mice for these antigrins, at baseline in orange, there is no modification of arterial stiffness at baseline. It is only in response to angiotensin II that we can observe a reduction of arterial stiffness in mice invalidated for alpha-1. That suggests that the lack of interaction between collagen and cell is able to control arterial stiffness. And this is the same if we look at the arterial wall hypertrophy in response to angiotensin II. The lack of these antigrins suppress the arterial wall hypertrophy in response to angiotensin II, and thus demonstrate that antigrins is involved not only in regulation of functional properties, but also in the structural modification of the arterial wall. And this has been also demonstrated in culture conditions. And if we look at another antigrins very well expressed in vascular smooth muscle cell, such as alpha V, beta 3 antigrins, again, at baseline, there is no modification, and even after angiotensin II, there is no modification in arterial stiffness. So the question at this time is to determine whether or not we need to interfere at the level of antigrin to change vascular remodeling, more particularly in vascular aging. In fact, we need to find the answer to these questions by looking at a more very complex structure, which is an adhesion structure between cell and extracellular matrix, and the name of this structure is focal adhesions. This is a dynamic structure which involves many molecules. And focal adhesion at base antigrin populations here in the membrane of the cell is in an inactive activation. There is no activations. In response to linkage with tallin, which is an intracytoplasmic molecules, there is a clustering activation and clustering of antigrin. And the linkage between tallin and vinculin produced a stronger clustering of antigrin, which is named molecular clutch. And we can see that all of this dynamic is very well regulated, and the half time is about few seconds to a few minutes. And they are characterized by association and dissociation between different molecules. So the complete clustering, activation of this structure between matrix and the intranuclear compartment of the cell define the molecular clutch, and the activation of this molecular clutch depends on the stiffness of the extracellular matrix. In the presence of a low stiffness of extracellular matrix, the linkage between tallin and actin is very low, and that's produced a slip bound in mechanotransductions, and the lifetime is very short. In a presence of very stiff extracellular matrix, there is a complete reorganization, and the linkage between tallin, vinculin, and actin are very strong, and the lifetime is a very long, more than few minutes. And these produce modification of proliferation and migration of vascular smooth muscle and cell into the intima. So at this time, the hypothesis is to determine whether the level of catch bound is able to control vascular remodeling during aging. In this slide, the focal adhesion are stained by vinculin. Vinculin is one of the most important molecules which compose focal adhesions. And in vascular smooth muscle cell, we can see the red staining of vinculin at the level of thoracic aorta, and in femoral artery, the size of focal adhesions is higher. And this is a good agreement with the arterial stiffness gradient we know along the arterial tree, because it is in the level of the thoracic aorta in which the elasticity is higher. and the elastic arteries is characterized by a low level of focal adhesions. So the higher level of the size of focal adhesions is associated with an increased arterial stiffness. But by interfering with focal adhesions, we also interfere with many signaling pathways within the vascular wall and within the vascular smooth muscle cell. And we should be able to interact either at the level of focal adhesions on the left side or at the level of contractility at the right side. And for example, if we look at focal adhesions, we know that there is an interaction between integrin in the membrane of the cell directly into the nucleus for expression of genes. And at the level of contractility, the most important pathway is the signaling of GPCR, J-protein coupled receptors. And J-protein coupled receptor activation is able to activate the RhoA kinase pathway, and RhoA kinase pathway is able to regulate arterial contractility. The key molecules is represented by myosin light chain, and myosin light chain here is phosphorylated by myosin light chain kinase and dephosphorylated by myosin light chain phosphatase. And the activation of RhoA kinase activity is able to reduce the level of myosin light chain phosphatase, thus increasing contractility. And we know, for example, that inhibition of GPCR, for example, the AT1 receptor for angiotensin II, is able to reduce the level of blood pressure in hypertension, and the reduction of blood pressure is associated with the reduction of arterial stiffness and a reduction in the outcome of cardiovascular event. There is another molecular pathway involved in the RhoA kinase activity. This is the role of culin-free molecules. Culin-free is able to inactivate RhoA kinase activity, and a mutation in culin-free in human or in mice is able to increase the level of RhoA kinase activity, and this increase in the activity of RhoA is able to increase the level of arterial stiffness in human and in mice. And even after angiotensin II administration and even after angiotensin II treatment, this process is accelerating. There is an acceleration of arterial stiffness in presence of culin-free mutations. So these demonstrate that downstream of GPCR, we are able to reduce and to modify vascular remodeling. So the first statement is that prevention of myosin activation decreases directly cytoskeletal tension and more than likely prevents focal adhesion growth, which decreases arterial stiffness and vascular remodeling. On a focal adhesion site, we can see here in vascular smooth muscle cell culture that lysophosphatidic acid, which is a myosin activator, is able to produce an increase in the size of focal adhesion in green here. And in the presence of an inhibitor of lysophosphatidic acid, PP2, administration in PP2 in vitro is able to reduce the number of focal adhesions. And the reduction in the number of focal adhesions is able to reduce arterial stiffness and to increase arterial elasticity. And in aortic rings, the same result was observed. So this is the second statement, that prevention of focal adhesion formations via FAK-SAK signaling inhibits stiffness development, because PP2 is an inhibitor of SAK family kinases. Sorry. So I would like to summarize the second part of this talk from signaling loop to arterial stiffening and vascular remodeling to clinical phenotype. The problem of all this summary is the is the redundancy, because the system is redundant, specifically the integrin family, which is the most important family of mechanotransductions. And in fact, the additive effect or synergistic effect utility need to be elucidated further. I would like, in the third part, to look at the influence of epigenetic modifications in the vascular remodeling. In this schematic, you can observe the normal life course of aging between childhood, early adult, middle age, advanced age, and the very old aging. And here, healthy conditions, elevated blood pressure, subclinical target organ damage, and cardiovascular disease. So in normal course, in blue, in advanced age, for example, there is occurrence of target damage in heart, in brain, in kidney, for example. In accelerated form of aging, which is named EVA, early vascular aging, which is here, with a red curve, there is a complete shift on the left. So at an early age, we can observe cardiovascular disease. But there is a recent concept which emerges a few years ago. This is an ideal life course, which is named SUPERNOVA, supernormal vascular aging. And there is 2 papers, and 1 paper is currently in press in hypertension. And we can see in this green curve that even in the presence of cardiovascular risk, there is no cardiovascular event even in the very old aging. So despite cardiovascular risk factors, supernormal vascular aging are protected against cardiovascular complications. And maybe this is at this type of occurrence of cardiovascular risk that we can observe epigenetic mechanism. We know that environment and genetic produced epigenetic modification leading to expression pattern changes and leading smooth muscle phenotypic switch. Few words about genetic. Genetic says, for example, that there is some SNP associated with cardiovascular disease. And for example, in the recent paper coming from the Aortic Gene Consortium, in a very large number of patients, more than many thousand people, they found identification of Wells locus as a determinant of arterial stiffness and vascular remodeling. This is relatively important because this locus is associated with regulation of actin polymerization and contractility and immune disorder. But finally, genetic failed to identify new molecular pathway involved in vascular remodeling and arterial stiffness, probably because of the multifactorial characteristic of the vascular remodeling and also because of epigenetic mechanism. So, epigenetic mechanism can be classified in different families. First, epigenetic programming produces histone and DNA changes, is characteristic by histone and DNA changes, leading to gene activation of repressions during development, repair, remodeling, and cardiovascular disease. And they are classified into different mechanisms: methylation, DNA methylation, chromatin remodeling, histone acetylation, and non-canonical RNA. And in non-canonical RNA, we can divide in microRNA, small microRNA, or long non-canonical RNA. And very nice, elegant illustrations of epigenetic modification. Here it is in atherosclerosis, in plaque. Here are vascular smooth muscle cells. And in plaque, in atherosclerosis, there is many cell populations. Normal cell population coming from the arterial wall, endothelial cells, smooth muscle cell, fibroblasts, for example, but also other extra vascular cells such as platelet, monocyte, macrophage, and so on. And one of the important result these recent years is the transdifferentiation of smooth muscle cell to macrophage-like cell. Smooth muscle cells are able to transform by themselves into inflammatory cell. And if we look at the signature of these epigenetic modification, we can see that smooth muscle cell in normal conditions is characterized by methylations and acetylations of histone 3 for methylation and acetylation at histone 4. And during these transformation, these transdifferentiations, smooth muscle cell keep the process of methylation, but acetylation is lost. And this is very important, and the techniques of cell lineage tracking is a very strong method to determine and to identify exactly which one of the population in extravascular wall or into the vascular wall able to determine this transdifferentiation. But we know now these macrophage cell light are coming to smooth muscle cell. Another example is the development of thoracic aneurysm, which is not an inflammatory molecule at the level of ascending aorta. And aneurysm is characterized by activation of TGF-beta and also SMAD2 overexpressions. And in normal conditions, in normal human aorta, we can see that SMAD2 is present in the cytoplasm area. And in normal conditions, the activation of SMAD2 is repressed by the MYC transcription factors. But in the presence of thoracic aneurysm, there is a shift from MYC repression to P53 activation of promoters of gene alpha smooth muscle actin and smooth muscle myosin heavy chain, producing expression of SMAD2. So there is a complete shift, and this shift is produced by acetylation, P53 activation, and acetylations. Concerning microRNA, there is different family of microRNA. Some of them are more characteristic of contractile smooth muscle genes, and other more characterized in proliferation and migrations. But to go further in the mechanism of microRNA, these group produced invalidation of the endonuclease dicer, which is a main processor factor of microRNA. And in invalidated mice for dicer, we observe a reduction of MIR26, MIR145, and MIR143, and this reduction is associated with a reduction of blood pressure and a complete disappearance of myogenic tone and vascular tone of the arterial wall. But, which is paradoxical, is that there is an increase in arterial stiffness. We are anticipated to have a decrease in arterial stiffness. In fact, we don't know exactly the mechanism. It was interpreted like an adaptive mechanism to limit the reduction in blood pressure. So the story is not over concerning microRNA and vascular remodeling. The first step to analyze mechanotransductions is to look first at the level of the plasma membrane in vascular smooth muscle cell, and to see that cell extracellular matrix are linked through the integrin to acto-myosin. We just looked at this before. But the second point is to look at the mechanotransductions at the nuclear envelope into the nucleus. And in fact, the mechanotransduction within the membrane of the cell and into the nucleus, that represents a continuum. There is a permanent link between mechanotransduction at these two sites. And into the nucleus, there is a complex, a dynamic complex between acto-myosin, the link complex, and the laminar scaffold. Briefly, at the inner membrane of the nucleus, there is a link between nesprin, in the outer part of the nucleus, with SUN proteins, into the nucleus membrane. And certain protein, via the cache domain, is able to interfere with nuclear lamine remodeling. And nuclear lamine remodeling, it is at this site in which epigenetic mechanism may occur in vascular remodeling. And how mechanotransductions in the nucleus works, it works via 3 different mechanisms: opening of chromatin under force, chromatin detachment, partial chromatin detachment, and partial lamine unfolding, promoting accessibility of transcription factors to the promoter of genes and interfere with the expression of genes. And one of the good example of this role of epigenetic is the syndrome of premature vascular aging in the progeria syndrome. Progeria syndrome is characterized by mutation in prelamine A, producing accumulation of progerin in the vascular wall, especially in stroke and peripheral vessels, in brain and peripheral vessel. And we can see here in the model of progeria the presence of progerin in green, and also the complete disappearance of vascular smooth muscle cell. And in these children, we can observe that pulse wave velocity, that the level of arterial stiffness is very high, 30 meters per second, which is much higher that this is normally observed in children at the similar age. This is a lot increase. And if we look at this slide, recently coming in 2017, reprogramming progeria fibroblast re-establishes a normal epigenetic landscape. For example, in control fibroblast here, there is, you can see methylation of fibroblast in green at histone 3, at lysine 4 and lysine 27. Sorry, I don't see very well. The lysine 4 and lysine 27. But in progeria, in fibroblast coming from progeria, there is a strong reduction of methylations. The nucleus are stained in blue. So it is able, by using the technique of IPS, induced pluripotent cell, it is possible to repair and to restore the methylation process, and which is remarkable, is to see in the same time the reduction of the progerin transcript in the progeria syndrome. So to look finally at the different curves of vascular aging, normal vascular aging, accelerated vascular aging, supernova vascular aging, we need to use models without hypertension, without atherosclerosis, and without vascular aging. And these models exist. This is a naked mole rat, which is the rodent who live the longest, almost 30 years old, sometimes 40 years old. And normal rodent, like mice and rat, normally lives 3 years. It doesn't look very fine, but in fact it is very kind, and in the hand it is very active, very active. It doesn't sleep anymore. So its weight is about 30 grams. It is a mice, but it is a rat. And some people have measured the level of arterial stiffness in this rodent. He lives in the East Africa in an underground environment. And they have a very long, very strong resistance to oxygen deprivations. They can live without oxygen around 18 minutes with oxygen 0. So we see here that arterial stiffness doesn't increase in these animals until 20 years old. And probably, maybe, this is one of the reasons why these animals do not die. And in fact, there is one explanation at this moment. Normally, there is the nuclear factor erythroid 2 related factors, NRF2, which is a molecule which the goal is to reduce the level of oxidative stress. And normally, this molecule is degraded by the proteasome. And in these particular animals, this degradation does not occur. And these may represent one of the mechanisms involved in cancer resistance, for example. And a Chinese group has recently demonstrated non-coding RNA in knockout mice. And they found a number of 40 non-coding RNA in naked mole rat involved in cancer resistance. But we don't know at this time which one of these non-coding RNA are involved in vascular remodeling and resistance to vascular remodeling. So I would like to conclude by this slide. There is a connection in terms of mechanobiology, which is the most feature of arterial remodeling, between hemodynamics, mechanosensing, and epigenetic. Arterial stiffness is just a marker of these interactions, and we need to consider integrin-based adhesions and nucleocytoskeletal coupling. And I would like to finish with this slide. This is an editorial of Martin Schwartz from Yale University in Science 2012. He proposed the concept of persistence and stability and disease, and he proposed an unifying principle. An unifying principle means that the treatment of vascular remodeling is common to common vascular disease. So by treating vascular remodeling, we are able to correct and to restore different vascular disease, from vascular aging to atherosclerosis, for example. And this involved in 3 different steps: extracellular matrix remodeling, leukocyte recruitment, and mesenchymal transitions with activation of TGF-beta signaling. And this is very interesting because this theory goes into the modern theory of personalized medicine. If by treating vascular remodeling by only one way, it's antagonist against this theory. So I would like to propose a selection of good papers to make a summary of these different topics. And I would like to thank my colleagues involved in this field. And if you are interested by vascular biology in this vascular disease, 2 postdoc positions are available in the lab. Thank you very much.
Thank you very much, Patrick, for this very exhaustive talk. Do you have any questions? Come on. So I can start, perhaps. I was wondering whether the type of chains in the integrins expressed by vascular smooth muscle cells could change upon time, and if alpha-1 could be predominant at one type and the alpha-7 could be then at another time, and so on.
Yes. We know, for example, that alpha-1 is expressed at a very young age in the maturation in vascular smooth muscle cell at the differentiated state. And during dedifferentiation, for example, alpha-V beta-3 is very well expressed, probably because of the modification of the capillary density, for example, and all of the immune disorder appear in the vascular world. So probably alpha-V beta-3 play an important role in fibrosis, for example. But because the system is redundant, it's very difficult to make a selective and to make a unique positions on integrin resistance, of integrin families.
And another one. And can the type of extracellular matrix determine the response? I mean, even though you have different type of chains expressed, perhaps the response inside will be directed by the engagement of these beta chains also, by laminin or collagen.
Yeah. There is more than 300 molecules in extracellular matrix. Some of them are like interstitial, like collagen and elastin, for example. But some are represented work like signaling molecules in the micro environment of vascular smooth muscle cell, like collagen 4, for example. And now the ultra microstructure of vascular smooth muscle cell may be very important in this field. And this is true at the individual level of vascular smooth muscle cell in the same locations. The microenvironment concerning matrix could be completely different in the same situations. But I think you know this better than me.
And so the question was motivated by another reason. How can we make the part about the stiffness dictated by the cells and that one that is relying on the type of matrix?
It's very difficult. In living cell, using atomic force microscopy, I think it is not the best method to see this. I think more sophisticated imaging techniques to see the dynamic of this process is able to see the migration, the modifications in the order of milliseconds. All the biological systems work in milliseconds, whatever the cell population and whatever the dedifferentiations. And sophisticated imaging techniques is probably the best method for the future.
Sorry, I just have an ad hoc announcement. So we introduce a new prize, which is a prize for the most active students who has the most meaningful questions, okay? Please.
So Josephina,
I'm not a student anymore. So thank you very much, Patrick. We will move to the next speaker.